Hello, everyone, and we are back for yet another exciting BenchTips webinar. I'm Tanuja Kopal, Consulting Editor for Biocompare, and today we are going to hear more about working with what are called as CAR cells. These are the chimeric antigen receptor, either T cells or natural killer cells, which are referred to as NK cells. So we are going to be hearing about CAR T cells or CAR NK cells. And these cells hold a lot of potential in terms of providing us with personalized medicine, particularly for cancer. But it's very tricky because these cells tend to be a little finicky. They need a lot of care, they need a lot of expertise when handled. So I'm very delighted to have three experts from different labs who routinely handle these CAR T and CAR NK cells to come here and be a part of this webinar. They're very willing to share their best practices, the technical know-how that they have learned over the years. So this webinar is really an opportunity for all of you to gain from their knowledge and hopefully save some time, cost, and effort on your part. And of course, you can always ask them any questions that you may have. So before I begin, I want to extend a big thank you to all our sponsors, Biological, BPS Bioscience, BioLegend, ACROBiosystems, Sartorius, and Biorad for their support of this webinar. Without that, it would have not been possible for us to bring you this webinar. They have also provided some resources that you can download from our resource center, so please feel free to check it out. So in terms of the format of this webinar, each of our three speakers will give short talks. And finally, after all the talks are done, we will have a Q&A session. You will be able to ask questions by typing it in the chat box, and you can do that by clicking on the Ask a Question button that you see on your screen. But please do not wait till the end to send your questions, because we typically get a lot of questions and we run out of time, and sometimes we are not able to get to all the questions that come in. So as you listen to the talks, and if a question comes to mind, just hit the Ask a Question button and we will get the chat message on our end. A few other things to point out, as I mentioned earlier, there's a resource tab. So just check it out. You have some useful information provided by our speakers and sponsors that can be downloaded. If you're having any technical difficulties, just click on Test Your Connection button that you can see at the bottom. That hopefully will provide the help that you need. You may also see some social media links that make it easy for you to share this webinar with your peers, colleagues, anyone you think who may be interested in hearing more about this topic. And finally, there's also a certificate of attendance. If you need one, it's there available for download. So with that, let me introduce our panelists. Our first speaker is Dr. Ismail Jan. He's a postdoctoral research fellow in the laboratory of Dr. Saad Kendarian, who is at the Mayo Clinic College of Medicine and Science. So Ismail has been studying senescence for a long time, and now he's looking at the role of senescence in CAR T cell failures. So he's going to start off the webinar providing some introduction to the topic, and then he'll go into some details about his work in this area. Our second speaker is Dr. Christopher Tor Sauter. He's an oncology fellow in the laboratory of Dr. Marco Ruella in the Center of Cellular Immunotherapies at University of Pennsylvania. So Tor is working on T cell cytotoxicity and how to overcome some of the mechanisms of tumor resistance. So he's here to share his work. And our final speaker today is Dr. Zhao Wang, who recently finished his postdoctoral training in the laboratory of Dr. Sandro Matosevich, a Department of Industrial and Physical Pharmacy at Purdue University. So Zhao has many years of experience in developing smart nano-based drug delivery systems and also in developing novel NK cell-based cancer immunotherapies. So his experience will be very valuable, as he's the only one who's specifically about NK cells for this webinar. So again, if you need more information about our speakers, their backgrounds, please click on the overview tab that you see on your screen. So I'm definitely very interested in hearing from all of them and looking forward to our discussion at the end of all the talks. So I'm going to just turn it over to our first speaker, Dr. Ismail Jan. Welcome, Ismail.
Thank you, Tanuja, for your nice introduction. So I'm happy to talk about what I do and talk about this really new, potentially curative therapy for cancer cells. And actually, it has many other applications. Soon, I think we will have additional talks about them. So as an immunologist, I really had difficulty to grasp the concept of chimeric antigen receptors. That's why, like, I needed to go back and understand a little bit more about the two most basic receptors in immunology: B cell receptor and T cell receptor. They share common structural similarities. When we look at it, we easily see that these receptors consist of two chains, and in the case of B cell receptor, light and heavy changes corresponds almost like alpha and beta chain in T cell receptor. In most of the cases, I see that this part of the B cell receptor is commonly compared like the T cell, whole T cell receptor. And even though it is not part of the T cell receptor, I also decided to show the CD3 zeta, which is independent but actually required to transmit the signal that is generated uh, by T cell receptor, because the intracellular domain of T cell receptors are not very strong and they cannot initiate the signal. And one other thing we notice is these both receptors, they have variable regions and constant regions. Variable regions are the ones that does the recognition. And among many other differences, one important difference between these two receptors that's also kind of relevant to us is while B cell receptors can recognize the target antigen in their native form, T cell receptors actually require some kind of processing of the antigen in the cells and presented to the T cells through MHC molecules. And now, if you look, this is a simplified version of uh, chimeric antigen receptor. Um, the first thing that strikes us is that everything is on this receptor. Everything is on this receptor is on a single chain. Uh, at the outer, this card structure is highly modular, which means like we can imagine like pieces of Lego came together and built this, which gives the advantage of actually removing pieces and modifying this structure even further for our purposes. At the outermost, what we see is the CFV, which is short for single chain variable fragment, and this part, which is responsible for recognizing the target antigen, it's structurally very similar to the B cell receptor part. And this actually helps CAR to recognize virtually any target cell, especially that's expressed on the surface of the cell. And now we see the transmembrane domain. This domain is not only anchors the receptor to the cell surface, but also transmits the signal. And at the innermost, now this CD3 as a domain, it is integrated into the chimeric antigen receptors. And now, just by looking this structure, we can tell that this CAR is second generation CAR. I'm mainly focusing on this because this version is the FDA approved chimeric antigen receptor. Here, the in the clinic, co-stimulatory, there are two alternative co-stimulatory domains for 1BB and CD28, and this really adds a lot of identity to the card that we use in the field. For 1BB is believed to give kind of like a milder activation response once it's activated with a longer duration. On the other hand, CD28 is the one that is known to give a stronger activation response with a shorter amount of time. And also, there are some studies suggesting CD28 also induces exhaustion phenotype in the T cells. And here I also wanted to show the how we generate the CAR T cell. So I also have these additional boxes to show the alternatives. And here, the bold ones are approved by the FDA, but the rest are also heavily tested in the clinic and preclinical models. So what we do is we obtain blood samples, peripheral blood samples, and then we isolate the PBMCs and further isolate in our lab, in our protocol, we further isolate the T cells. And if you're like me, who spend most of your career culturing cancer cells, you probably don't realize the luxury of having an unlimited source of cells. Easy to modify, you can modify, select, because cancer cells can outgrow. They are like they're enduring kind of selection, they overpopulate. But like one thing about T cells, we can consider them as almost like shy. If they don't have reason to be around, they won't. We will just lose them. They will disappear. In order to convince them to stick around, we actually have to stimulate the T cells. And in our protocol, we choose like CD328 dynabeats, which not only activates the T cells, but also prepares them for the next step, which is the transduction. And in our protocol, after day of stimulation, we introduce the lentiviral particles that contains the chimeric antigen receptor. And then we maintain this culture for up to like 6 days. And then we keep the cells happy, like fresh media, lots of source and nutrients for them. And at day 6, we take advantage of these dyna beads. With the help of strong magnet, we remove the beads and then just purify the cells, and then we rest the cells for 2 days. At the end of the 8 days, they are ready to either to be used, or we can just freeze and store for like any other cells. We can freeze them with protective agents and store them almost like anytime that we want. So there are some challenges associated with this CAR T cell treatment, but before I complain about them, I just wanted to point how efficient this treatment is. So here on this graph, what we are seeing, the historical survival rate of the relapsed and refractory ALL patients. These are the patients nowadays can take advantage of the CAR T cell treatment. And if you notice, the 1-year survival is below like almost like 25%. And now with the introduction of chimeric antigen receptor T cell therapies, we see that this almost moves up to like almost 80%. So that's great. The durable the response rate is almost like 80%, and 60% of the patients that are treated with chimeric antigen T cell receptor therapies are achieving the complete response. But however, like in time, you will see that this is decreasing steadily. But then if you look at like the challenges, the initial challenges is the cytokine release syndrome and neurotoxicity. Good thing is they're mostly reversible. Unlike the other treatments, for example, chemotherapy. If you use chemotherapy, induce like DNA damage or something, they are like almost permanent. And the other thing is only a few subset of cancer patients, mostly B-cell related malignancies, can take advantage, even though there are some promising studies happening in additional diseases, especially like the solid tumors. We still don't have FDA approved in any other cancers yet. And also, as I just mentioned, some patients, like T cells are not good enough in a way that like you cannot produce functioning active CAR T cells out of them. So there are some failures there, and also there are failures in the long term, which is also my main interest. Like I'm trying to understand why some patients respond for a while and then stop, and why others they still have continuous response. So what I mean about like the CAR T cell failure in the long term, imagine that this is the timeline, and we have establishment of the malignancy here, and we introduce the CAR T cells. What they do is they traffic to the site of tumors, and they recognize their targets, and this kind of induces some activation response in the CAR T cells. They expand, and then they start showing cytotoxicity, starting to eliminate the target cells. This leads to remission. But we know that cancer cells are known to relapse. They can come back, and in patients, these recruit CAR T cells again because it's in their system. And depending on the patients, this cycle happens. For some patients, it is like higher numbers, or for patients, something happens along this activation, either there's a change in the tumor, which could be like losing the antigen, or also something happens with the CAR T cells that like they are not functioning anymore. In fact, almost like 47% of the time, the relapsed clones, they still have the targeted antigen. And also there is actually one additional method here that I'm not mentioning because in the next section we will talk. It's a very interesting concept. And this all leads to failure of the CAR T cell treatment. And if we look at it, we have options. One option is like we can try to improve our CAR T cell fitness, or we can also try to define some novel targets, even though most of the time we focus on CD19 targeting CAR T cells. There are some additionals that are being tested, like CD20, 22, some others, heavier light chain targets from B cell receptor. And so then we really understand like the role of impaired T cell fates in the CAR T cell failure. And so here, what we are seeing in this simple cartoon, the minimum interactions that is required for proper T cell activation. One is through T cell receptor complex and the MHC on the target cell, and the other thing is the co-stimulation. And after this, we have three possibilities that may occur: anergy, exhaustion, and senescence. Anergy is not a big problem since the introduction of the second generation of CAR, but the main point is that all these three fates have the same or similar consequence. It's just failure or suboptimal CAR or T cell activity, which leads to the failure. And when we look at the relationship between these fates, we know that normal anergic and exhausted cells are thought to dynamically change. A cell can go into those states and come out of it, but the senescence is considered to be mostly like a permanent cell fate. Once it is developed, cell stays will that way. And now in my research, what we are trying to understand is if this is the failure, we don't know where this failure falls. Is it because of one of these three possible fates, or it is just a combination? And that's what we are trying to understand. But before that, I would like to talk a little bit more about exhaustion and senescence. Uh, Both of them, as I said, are like giving like this similar result, which is the suboptimal activity. And they also have additional common features. For example, this table from the review is nicely summarizes the differences or similarities. And you can also notice that, for example, TIM3 is associated with both sides. It will come to that. That's why I'm highlighting that part. And again, one very big difference that we are also trying to utilize in our experiment is that exhaustion is reversible while senescence is highly permanent. Just in order to utilize this, we developed this an activation resting cycle to study the CAR T cell dysfunction in vitro. The first part is there are two arms associated with our study. One is the CAR T cell production, which takes almost like 8 days. Excuse me. And then we also have activation cycle. In this one, we co-culture our CAR T cells with a cell line that's positive for the intended antigen, which is the CAR T cell, which is the JECO one, that's CD19 positive. We culture them and then rest, culture and then rest. Now, what we did is we are mimicking the relapse of cancer, and also we are giving cells to recover from exhaustion to look beyond if there is a senescence happening in our cells. And these are the two main assays that they apply, especially in vitro, proliferation and cytotoxicity assay. These two assays are also based on co-culturing our CAR T cells with the target cell. We use JECO1. In the proliferation, we just co-culture them and then we count. And in cytotoxicity assay, we co-culture them and we measure the bioluminescence because we transduce our target cells with luciferase, when we give the substrate, we measure the bioluminescence, and based on the signal lost with the mixture of the CAR T and target cells versus just the target cells alone, it tells us the cytotoxicity levels of our CAR T cells. And with that, so we collected some blood samples from normal donors and applied them through the activation cycle. So day 8 are the CAR T cells that are the equivalent of the clinical use. After one activation cycle, we have day 15, and then another activation cycle, we have day 22. When we look at the individual patients, this proliferation data and cytotoxicity, different effector to target ratio, and of the different CAR T cells that we have, BBZ and 28Z. So then we combined multiple donors, and what we noticed is the CAR T cells that are co-stimulated by 4-1BB seems more like a stable proliferation response, but we see loss of this proliferation in the 28Z or the CAR T cells that are co-stimulated by 28Z. And for cytotoxicity, it seems like both of them are kind of affected. There's an initial increase for the BBZ, but then at day 22, there is not increase anymore. And there's an obvious decrease in the 28Z. And we also monitor the immunophenotype of these cells. And what we initially checked the exhaustion markers. And what we noticed is there wasn't a very unified increase in the exhaustion markers, suggesting that the cells, when we put to our protocols, we don't see the cells were not showing sign of, at least immunophenotypical sign of exhaustion, except for actually TIM3. TIM3 went up and stayed up. And if you remember, TIM3 expression was also associated with the senescence. And then we decided to look at the senescence marker expression. We checked this with flow by doing intracellular staining. And we also saw that there's an increase in the level of P53 in both of the CAR T cells that we tested. And there's also another senescence marker P16 increase in, especially in the BBZ. So we'll try to understand this more. But with that, I would like to summarize what I showed. So we know that CAR is designed based on both BCR and TCR, just carries features of both of them. And we know that CAR T cells are very impressive in the clinic, but there is still like further improvement that we can actually do if we define the shortcomings. And this could turn CAR T cell treatment as almost like a potentially curative therapy option for cancers. We know that exhaustion is heavily studied in CAR T cell field for a good reason, because exhaustion is an important and a common phenotype for T cells. But we also realize that there is also a need for studying additional T cell fates, kind of like almost like scratching the surface to see what is behind. And that's why we develop our activation and resting model to study CAR T cell failure. And now when we apply this recurrent activation, we realize like there are some changes that are consistent with senescent T cell phenotype. With that, I would like to thank my lab, T cell engineering lab at Mayo Clinic, especially my mentor, Dr. Kandarian. I also would like to acknowledge our funding agencies, especially Regenerative Medicine, which is supporting majority of my work.
Great. Thanks, Ismail, for the great talk, and thank you, Tanuja, for organizing this. So again, my name is Tor. I'm in the Rowell Lab at the University of Pennsylvania, and I'm going to review a little bit about our approach to the manufacturing and preclinical evaluation of novel CAR T cells. Slides not advancing. There it goes. I have no disclosures. And for a brief outline for my talk, I'll review the barriers to clinical CAR T-cell function, and then discuss some of the methods available for the preclinical evaluation of novel CAR T-cells. And then finally, I'll discuss some of the technical considerations in the manufacturing of CAR T-cells that can influence validity of results. So just to review briefly, CD19-directed CAR T-cells are highly effective in the treatment of relapsed B-cell ALL and some mature B-cell lymphomas. However, most patients will ultimately relapse with their disease. To understand CAR T-cell failure, many groups, including our own, have described a variety of parameters associated with failure. And this cartoon summarizes some of those variables. A major focus of our work is on the understanding of tumor resistance to immune-mediated killing, for which we are developing novel CARs to overcome this phenomenon. Methods to evaluate CAR T-cell function uniformly start off with stimulating a T-cell via the CAR receptor using the target antigen. This can be done using antigen bound to an aggregate or using cells expressing a target antigen. Or to understand more complex environments, physiologic stresses can be done using organoids or in vivo model systems. In general, the output of these methods examines T-cell alterations such as phenotype, exhaustion, tumor cell killing, proliferation, cytokine secretion, and some additional parameters in vivo that I'll discuss later. This table from a review summarizes some of the in vitro assays available to assess T-cell behavior. Given our work focuses on tumor resistance to T-cell killing, we seek to measure effector function of CAR T-cells. Traditionally, surrogate markers such as cytokine secretion were heavily used to demonstrate CAR T-cell activity. However, we tend more towards direct cytotoxicity measurements over surrogate markers. And for this, there are a variety of assays available. While in vitro assays are invaluable because of their ease of use and high throughput nature, how CAR T-cell in vitro behavior predicts in vivo and clinical behavior is not clearly established. This table summarizes types of assays that directly measure T-cell mediated cytotoxicity. Traditionally, chromium release assays have been the gold standard. However, most labs have moved away from radioactive assays. Our lab tends to heavily use bioluminescence luciferase-based assays, which use tumors with a luciferase reporter gene. Alternatives include impedance-based assays using the Excelligent system. However, this is optimal for adherent-based tumor cell lines over non-adherent target tumors. Alternatively, flow cytometry-based killing assays are useful when one desires to measure early markers of apoptosis or when the target tumor lacks a reporter assay. Additionally, not shown in this table are fluorescence-based high-throughput microscopy systems that can be used in an incubator, such as the IncuSite or CellSite platforms. This table highlights a variety of in vitro luciferin-based cytotoxicity assays using different effector cells. And I'd just like to highlight here that there's a wide range of conditions depending on which model system is used. So there's a wide range in the number of target cells that are placed into the assay, of effector to target ratios, and incubation times. So this really requires familiarity with the assay and model system you're using. I'll also comment that effector to target ratios may be different between CAR T cells and TCR-based cellular products, if that's your area of interest. So just to kind of briefly review how these are set up. In this example, we use NOM6 with the luciferase reporter that's plated onto a 96-well plate at different ratios with CAR T cells. And this can be analyzed at different time points, typically between 3 to 4 days. This is quantified, and then we can qualitatively compare the rate of tumor killing between different novel CAR T cell products compared to a control. We make use of a fluorescence-based system to extend the length of our tumor killing assays, sometimes out to a month. So in this example, we use the CellSite X platform that sits in our incubator. And using this system, we can compare tumor growth by GFP intensity over time on a logarithmic scale. And we see that different candidate preclinical CAR T cells have enhanced tumor control over a longer time frame that would have been possible with a luciferase-based assay. Once in vitro efficacy is demonstrated, the gold standard for demonstrating efficacy remains in vivo model systems, of which there are many. Our lab heavily relies on immune-compromised model systems using xenografts, either cell line or patient-derived xenografts. So there's many different types of model systems for this. Additionally, there's immune-competent model systems that are useful for describing interactions with the tumor microenvironment, and some other types of in vivo systems that are more useful for describing toxicity evaluations, such as modeling cytokine release syndrome. However, there's no gold standard for the evaluation of CAR T cell toxicity, and that remains a big barrier in the field. Additionally, the in vivo model systems are extremely disease-oriented, so we focus on leukemia and lymphoma models, but there are also solid tumor models available to us. So just for example, in this schema of how these are typically performed, immune-compromised mice such as NSG mice are engrafted with a tumor, typically luciferase-expressing tumors. The mice are then monitored for tumor growth, and then at a pre-specified tumor burden, mice are treated with CAR T cells. The output from these experiments are survival, tumor growth, as measured by luciferase measurements using this type of bioluminescence imager, as well as CAR T cell proliferation and phenotype by flow cytometry from peripheral blood. Sometimes we also do more advanced assays such as single cell sequencing. So for example, we have a candidate knockout CAR T cell with a knockout in a gene of interest compared to a standard CAR-19 here in blue. And when we examined for cytotoxicity using a bioluminescent in vitro assay, we see really identical levels of cytotoxicity during this short course in vitro assay. However, when we examine the performance of these CAR T cells in an in vivo model system, we see that our knockout CAR T cell has enhanced tumor control compared to a control CAR T cell, likely indicating that in vivo models introduce physiologic variables not present in vitro. And this really reinforces why in vivo model systems are essential in the preclinical evaluation of CAR T cells. So I'm going to shift briefly into talking a little bit about CAR T manufacturing and really highlight some of the issues that can unintentionally influence results. And I know Ismail talked a little bit about this, and I'm going to talk a little bit more in detail about what our experience has been. So this is a general schema for CAR T cell manufacturing, including CRISPR editing. So primary T cells are taken from a donor, electroporated with Cas9 and guide RNA. As Ismail described, they're stimulated using CD3, CD28 beads, although there are other methods as well, and then transduced for the CAR using a lentiviral transduction system. And then expanded, the beads are typically removed at day 5 or 6, and then our expansion protocols can go all the way up to day 16 or 18 before the CAR T cells are frozen down. During this time, we take particular attention to monitor population doubling, as well as cell volume. So as T cells are activated by the stimulation beads, they really increase in size, and then following removal of the beads, they begin to rest down. Oops. Activated T cells have enhanced tumor killing properties and other behavior. So it's really essential to ensure CAR T cell uniformity in our assays. So we monitor size to ensure that the T cells have truly rested down by the end of manufacturing, so that we can compare them accurately in assays. Some other variables that can change your T cell function during manufacturing include duration of T cell expansion, selection of culture media and serums, and then the use of exogenous cytokines. So in this review highlights how changes in serum and media may change T cell behavior. So that includes altering T cell activity in vivo and changing gene expression systems or gene expression profiles. The use of exogenous cytokines is fairly routine. So interleukins 2, 7, and 15 can enhance T cell proliferation and essentially make your manufacturing processes more cost effective. However, this changes T cell behavior. So on the right here, we see some notes about what has been described as consequences of using exogenous T cells. So increasing anti-tumor activity, decreasing apoptosis, but also potentially increasing Treg expansion and exhaustion. So this is not necessarily a bad thing, but should be standardized and reported in a methods section. So here's just an example of kind of the consequences that I've described. So in this experiment, we compared a long manufacturing protocol to a short manufacturing protocol. So a long manufacturing protocol, again, has the advantage of producing more T cells, while short manufacturing protocol could potentially deliver a clinical product to a patient faster. And here we monitor the population doublings. When we examined in vivo the comparison of these CAR T cells produced via long and short protocols, we can see that CAR T cells manufactured using a short protocol demonstrate enhanced tumor control compared to the long protocol. So we have tumor flux in red, tumor flux on the y-axis with the short protocol in red here. So this is thought to be due to enhanced proliferative capacity and cytotoxic functionality of a more naive T cell phenotype that's produced during a short manufacturing process, compared to a long manufacturing process, which may produce more terminally differentiated effector T cells with poor proliferative capacity that are more prone to exhaustion. And this has been repeated by other groups and published upon. So to summarize just a few key takeaways, there's multiple in vitro assays for the preclinical evaluation of CAR T cells that can look at a variety of parameters. Demonstrating in vivo anti-tumor efficacy remains the gold standard for the preclinical evaluation of CAR T cells. Predicting CAR T cell in vivo and clinical behavior using in vitro assays remains imperfect, unfortunately. And then standardizing CAR T cell manufacturing will really enhance the validity of your results if you're doing this kind of work. So with that, I just want to thank my group, my mentor, Marco Ruella, and of course, Tanuja for organizing this webinar.
All right. So first, thank you having me on this webinar to talk about some science related to the NK cells. So the title of my talk today is Reprogramming Human NK Cells or CAR NK Cells for Cancer Immunotherapy. So I will start from the first part. I will give a brief introduction of the NK cells, including NK cell basic biology, their effector mechanism, and when compared with CAR T cells, what is their pros and cons? So what are NK cells? So they are cytotoxic or lymphocyte of the innate immune system that can direct kill abnormal cells, such as virus-infected or cancer cells. So their mediated killing are prior stimulation non-required, antigen non-specific, and HLA non-restricted. So how these NK cells recognize and finally kill the target cells? Basically, these NK cells express a series of germline encoded inhibitory and activation receptors. So to kill or not, it depends on the balance of these two sides. For example, many cancer cells downregulate MHC1 molecule to avoid cytotoxic or T cell mediated killing. In parallel, they upregulate stimulatory ligands for NK cell activation receptors, such as NKG2D. So this will break the balance and lead to the NK cell activation. Once NK cells get activated, they will one side secrete perforin and granzymes, just like T cells, which will lead to direct lysis of the cancer cells. Another side, they also will release some cytokines, such as IL-1 gamma and TNF-alpha, which enables them to further crosstalk with other immune cells and further to shape the adaptive immunity. So compared with CAR T cells, CAR NK cell therapy really have some unique advantages. First, they can be used as an allogeneic product. Second, they are pretty safe, have not shown obvious cytotoxicity or some side effect in the clinical trial. The third, they have multiple mechanisms to recognize the target cells, not only dependent on the card structure. Finally, they have ADCC activity, which mainly due to the CD16 expression. Of course, these NK cells compared to T cells also has some disadvantages, includes limited in vivo persistence and relatively low viral transduction efficiency. So second part, I will give you an overview of NK cell therapy pipeline, such as NK cell sources, how to isolate NK cells, and cell culture and expansion. Final is genetic modification. So where these NK cells come from? NK cells can be obtained from a broad range of sources, such as peripheral blood and cord blood, stem cell derived, and some immobilized NK cell line. So we commonly in the lab or clinical isolate these NK cells from peripheral blood or cord blood from either positive selection or negative selection. Basically, these selection kits are widely available in different companies. And when we isolate these NK cells, we typically culture these NK cells either by using feed-free condition and feed cells-based condition. Okay, to summarize, we have a lot of different kind of feed-free medium available, such as NK Max medium, CDS expanded medium, and immunocult NK cell expansion kit from stem cell. And for feeder cells, there are two typical widely used feeder cells. One is engineered K562 cells with either membrane bound I-15 or I-21 plus 4-1BBL. Another is EBV-LCL, which basically is a virus, transformed B cells. And also, besides this, culture system, we also can culture these NK cells in different, instruments such as GRAC product, so that can further help we expand these NK cells. So then, regarding the genetic engineering, so broadly there is two main approaches. One is virus-based, another is non-virus-based. Regarding the viral-based, typically there is two big categories. One is lentivirus or retrovirus with different envelope. Okay, this virus will induce some non-specific, randomly integrate into the NK cell genome. Then later on, the scientists or people realize we can use adeno-associated virus plus CRISPR-Cas9 technology to induce some specific knocking, while it is limited by the payload size, which is around 4.7 kilobase. Regarding the non-virus-based genetic knock-in, there is a couple ways, such as mRNA, which we induce some transient knock-in, and transposons such as piggyback, Sleeping Beauty, or TC Buster systems, will induce some large payload knock-in, but still non-specific. So finally, is different non-viral vectors, such as double-strand DNA, linear single-strand DNA, and circular single-strand DNA, plus CRISPR-Cas9 technology, which can give us specific locus knock-in. So the final part, I will take my own research as an example to further discuss the potential application of NK cells in the cancer immunotherapy field. So first, I give a slide to summarize, what's the challenges of NK cells in tumors, especially for solid tumors. So how these solid tumors make NK cells invisible? So typically, there's three strategies. First is antigen escape due to highly tumor heterogeneity. The second is dysfunction, due to severely immunosuppressive tumor microenvironment, such as tumor cells secrete a lot of soluble factors, including TGF-beta, adenosine. Also, meantime, they downregulate the activation receptors, such as NKG2D, CD16. The final part is poor infiltration and penetration due to the specific, tumor size. So here, what we try to target is a solid tumor called glioblastoma, which briefly called GBM. It is a #1 aggressive and malignant primary brain cancer. And also, it is highly infiltrative and heterogeneous, which made it very resistant to different therapy. And during past 16 years, only one new drug was added into the market, and with limited benefit. So we think NK cells maybe can utilize as a therapy to overcome or to treat this type of aggressive cancer. So here is our strategy. So we built a multifunctional construct to enable to NK cells express multiple arms, so targeting these 3 different pathways. As shown here, as shown here, the first part and the second part is synthetic chimerical antigen receptors, including GD2 CAR and NKG2D CAR, which can enable NK cells to overcome the antigen escape. The second part is a tumor-responsible, cleavable CD73 SCAV part. So just think about this NK cells, when they get into the tumor site, this part will release so that they can bind to the CD73 on the tumor cells, so that blocking the adenosine production. So we can overcome this immunosuppressive tumor microenvironment. So to increase infiltration of the NK cells, here we introduce another part, which is a small molecule treatment, which called chloroquine, which really can target the autophagy pathway in tumor in GBM, so that they can increase the NK cells infiltration. So how we generate this, multifunctional CAR NK cells? So we get whole blood from the healthy donor, and then we isolate NK cells from this PBMC through the negative selection kit. Then we expand the NK cells around 1 week. Finally, we use lentiviral transduction methods to do the transduction. So from the bottom panel, you can see the isolated and expanded NK cells are highly pure, so which we usually use CD56 positive and CD3 negative as a phenotyping. And also we can achieve a decent construct expression, both NKG2D and GD2 CAR, and also CD73 SCAV part. And also these engineered NK cells can be well expanded, so that we can utilize these NK cells to further evaluate the in vitro and in vivo anti-tumor function. So then we launched a series in vitro assays, including cytotoxicity, degranulation, cytokine production, as well as CD73 activation, to really evaluate the in vitro anti-tumor activity. Besides, we also further look into the exhaustion and off-target effects. So regarding the infiltration part, so we use this small molecule called chloroquine. So we can see here, this chloroquine treatment can inhibit, just like NK cells, partially inhibit the tumor growth, both in vitro and in vivo. Alongside this inhibition, they can boost tumor cells to secrete CCL5 and CXCL10, these chemokines. And we built a in vitro BBB model, so we can see this CCL5 and CXCL10 really can bring NK cells across the BBB, so that we can achieve this increased penetration and infiltration. So finally, we built an intracranial mouse model, and we treated these mice, with 3 weeks treatment plan, and we give continuous 3 days of this small molecule. Then, following that, we give an intracranial injection of NK cells. So from this result, we can see the chloroquine treatment plus these multiple functional NK cells can really show significant tumor growth delay as well as overall survival extension. So, through all the above, slides, I hopefully introduce what is NK cells, how they kill, how they kill the target cells, how we commonly evaluate their anti-tumor killing ability. Final slides, I just want to summarize, give everybody a broader view. So in future, how we to improve this NK cells mediated therapy. So I will briefly introduce through the 4 parts. First is genetic engineering. As everybody know, now the genetic engineering technology really developing very fast, such as CRISPR, mediated base editing and prime editing. So we can utilize these technologies to really achieve high on target or low off target genetic engineering efficiency. The second part is still how to really potentially overcoming this tumor immunosuppressive environment, especially solid tumor cells. So we can build a different strategy to target multiple checkpoints inhibitors. We can do multiple knockout and knock-in to make these NK cells more functional. The third part is manufacturing and the delivery part. How to refine methods, to increase the NK cell, expansion, production, and also cryopreservation, and then optimize a delivery system. So which finally make these NK cells available to more patients in an economic way. So finally, how to broaden the applications. So expanding the scope of the NK cell therapy, not only cancer, and we realize NK cells can utilize as a potential treatment in other diseases, such as autoimmune disease, infectious disease, and neurodegenerative disease. So then I will, just try to acknowledge all the members in the, metastatic group. Even as Tanja says, I just finished my postdoc. Now I'm, moved to the industry, but I'm still want to, give them, the sum up, applaud for all the support. And again, thanks for all your attention. And I would like to have any, answer any questions in the Q&A session. Thank you again.
Thank you, Joe. Thank you, Ismail and Tor. Really enjoyed your presentations. Quick reminder to all our attendees that you can send in your questions by typing it in the Ask a Question box that you see on your screen. Also wanted to remind you that the webinar is available on demand, and a link to the on-demand version will be emailed to all of you within 24 hours. And it will be on Biocompare's website, which is biocompare.com. So with that, let's come to some of the questions that have come in. Oh, also a quick reminder that we do have a resource center. So definitely click on the resource center, because you will see a bunch of resources provided by our sponsors, and you will also be able to download some of them for future reference. All right. Let's go to some of the questions now. First, let's start by talking about, this is for people who are, I guess, new to the CAR-T, CAR-NK cells field. So I don't know who would want to take this question first, but what are some of the common mistakes that people who are new to the field make? So would any of you like to share your experiences when you first started working in this field? What worked, what didn't work, where did you, what were some of the things that you ran into? I'm sure the audience will benefit in listening to some of your experiences. Tor, did you want to start first?
I think, again, my presentation, we're kind of comparing a lot of different types of CAR T cells. And I talked a lot about the manufacturing process, and I think, there's a lot of work in the industry broadly about, what are the ideal manufacturing processes. In our work, I think one of the things that I ran into and didn't really kind of think about this initially is, we compare all these CAR T cells head to head, and we try to normalize as much as possible, using the same E to T ratios. But when you manufacture them, inevitably there'll be differences in essentially the transduction efficiency, or basically the surface CAR expression, kind kind of on your different types of constructs. And I think minute differences are fine, but if the transduction efficiency is different than by more than 5 or 10%, that really needs to be adjusted for by essentially using untransduced T cells to create a situation where your transduction efficiency is equivalent across all of your conditions. So if you're making, many different viruses, it's really hard at times to get the transduction efficiency equivalent, although we do our best to do that. So that's kind of one kind of a very, very technical thing that people may run into that I didn't talk about. I'm not sure if any of the other speakers have other things they can comment on.
Yeah, I can contribute a little bit. So especially in the research side, of course, we like to have our results tested with multiple donors, like coming from different individuals. But most of the time they are not matched. They could be very different. So assumption of like a data generated from one donor may not match to the others. So those are like the considerations I think when we set up our experiment. Sometimes like I start certain amount of cells with a donor and assume other T cells from other donors would just like be similar, but sometimes they are not. And also, again, in the research side, we are taking usually like T cells from healthy donors. But then when we start setting up experiments from some clinical samples, some patient cells, that also kind of don't go well because like the proliferation or activity is usually lower with the clinical samples. Like I was thinking that those could be like some important considerations.
Definitely. So you think using the autologous cells, like the off-the-shelf cells, will actually help with some of these issues?
I think like there is. I think one of the big thing in the CAR T cell field is to be able to develop a T cell that would be on the shelf, like you said, so we can do not autologous but allogenic, and I think that would really improve activity a lot because the T cells from patients, they are they're interacting with cancer cells, and cancer cells have this heavy immune suppressive phenotype to the immune cells in general. And on top of that, now CAR T cell treatment is approved for second or third line of therapy, which means like the patients are going through several rounds of treatments, including chemotherapy options. And this also, I think, affects the T cell activity, which we use it to generate CAR T cell. But like you said, if the allogenic was an option, the problem with the allogenic is two levels. Like either these cells that are taken from some other individual transplanted to another may attack other cells in the system, or also the other immune cells in the system may attack these like graft versus host versus graft. So there are some promising results, but the challenge is to just protect the cells from immune attack and also prevent our CAR T cells attacking, because they will still have their intact CD3 like TCR complexes.
Right. Yeah. Thank you for correcting me about the allogenic. Yeah, I always get confused between the two. That is a good point. I wanted to bring Giles' perspective in because he doesn't have anything to do with the CAR T cells at all. What is your experience like with the NK cells, Giles, and also any advice for people who are just getting into the NK cells? Tor, you talked a lot about the novel CAR T therapies that that people are now developing. So is the focus in the novel therapies all around improving exhaustion, or are there other approaches that these novel therapies are also going?
So the answer is no. There are a number of CAR T cells that are, being tested in the clinic right now that do a couple of different things. Certainly, PD-1 knockout and TIGIT knockout CAR T cells have been developed to avoid exhaustion, and then changing manufacturing processes to avoid exhaustion is one approach. Other strategies that are being used include CAR T cells that can basically essentially modify the tumor microenvironment by secreting cytokines that can recruit other immunologically active cells. I think what my project and our group is trying to do is kind of even a third option and enhance the intrinsic ability of T cells, the intrinsic cytotoxic ability of CAR T cells to make them basically like super physiologically cytotoxic, and hopefully, kind of with the hope that that strategy can be broadly applied across different types of diseases. There's also a variety of other kind of approaches as well. I'll say that people have tried using checkpoint inhibitors in combination with CAR T cells with limited utility. So I'm not sure. We'll see. I think there's going to be some data coming out. How modifying the surface exhaustion markers or using checkpoint inhibitors pans out, but I'm not sure that's going to be the full answer for improving outcomes.
Sure. And then the last question is, you're doing all this work in vitro models. How does that really, and you're extrapolating these results, assuming that they'll translate well into the clinic in patients. What are your concerns about that, and how are you guys addressing that?
Yeah, so you're asking like how in vivo or in vitro data reflects the in vivo, right?
Correct.
Yeah. So I think Tor mentioned this a little bit during his presentation. Yes, there is some like the advantage of in vitro testing is it is more practical, easy to do, and you can collect a lot of data in one experiment. And a lot of times I think our results agree, but it doesn't mean that in every experiment like that will be very parallel with in vivo. And then in vivo is obviously closer to the clinic, but then also I think I'm taking this part from Tor. Our in vivo, the most commonly used in vivo system would be like the immune incompetent mice, like NSG mice or something, and so those mice are also have some challenges, inherent challenges with them, and that also may not really reflect what will happen in the clinic. I'm not saying like this is highly possible and it's highly common, but there are we always go from stepwise, like starting from in vitro and then go in vivo. If it works, then I just go to the next step. Unfortunately, as of now, I don't know if we have like a perfect system developed to test our findings.
Yes, no, completely understand. But how? So if you want to elaborate on that, but how much do you really trust the data that comes out of your in vitro models and your in vivo mice models? Are they fairly translatable?
Yeah, that's a really good question, really hard, and I think the answer is just depends. It depends on what disease you're talking about. I think our solid tumor models, especially I think in the immuno-incompetent mouse models are lacking, and this is, I really think because we know that the tumor microenvironment is highly immune suppressive. I mean, in liquid malignancies, sure, but also like particularly in solid tumor malignancies, and that's one of the reasons we think CAR T cells do not work as well in this situation, and you just can't get that tumor microenvironment that is reflective of human biology in immunocompetent models. I think we're a little bit more confident, with some of our B-ALL models, but still, I think you're never really going to know until you bring it to clinic. So we do this stepwise approach through in vitro, and then use our best available mouse models. And then again, I think I talked about this during my talk. None of these models really are well validated for toxicity evaluations. So that's why I think doing a really good, really safe phase 1 clinical trial is important, especially when you're talking about adding cytokines or changing signaling or reducing exhaustion. You really might not know what's going to happen.
Thank you so much. All right, so I just wanted to thank you all again for such wonderful talks. Thank you for joining the Q&A and sharing your ideas and your insights. So with that, I was just looking at the question box. So there is some question about the certificate of attendance. Yes, definitely we do have a certificate of attendance available that you can download from our website. And also, as I mentioned earlier, the webinar will be on demand in about 24 hours. So please check our website, biocompare.com, and you'll also receive a link to the archive by email. So with that, I would once again like to thank our panelists, and also a big thank you to all our sponsors, Biological, BPS Bioscience, BioLegend, ACROBiosystems, Sartorius, and Biorad for supporting this BenchTip webinar, and to the team at Biocompare for their help in putting this together. I think there are no more questions. So with that, we will end the webinar. Thank you all for joining me today, and we look forward to seeing you at the next event. Goodbye.